Now we dive into the analytical assays that, of course, go hand in hand with our workflows to generate engineered immune cells. A short overview what we will be talking about. So first of all, very quick introduction. Then I would like to dive into the analysis of the tumor microenvironment, very important part, of course. Then the phenotyping done via flow cytometry, which is also key in a lot of different assays. Then last but not least, functional assays. This is also a hot topic currently. Good. Then let's get started. So this is something we've already seen in the first part, just to kind of quickly review. So there's a huge pipeline for cancer cell therapy products coming up. So a huge amount of new products being in preclinical phase and also in clinical phases. However, when you think about how many made it already onto the market, it's just 6. Yeah, you can say it's just 6 or already 6 made it. But what it actually says is that there is quite a bottleneck. So there are a lot of projects ongoing, and it is not always a success story. And one of the big hurdles definitely are the analytics. So the analytical assays that go along with IPC-QC monitoring, but also in earlier phases. So as already mentioned, analytics, they are really needed in all different phases. And I think this graph we have also already seen in the first part. So we have different phases that eventually hopefully lead to a commercial product. And we start with a discovery phase with target identification, validation, then lead identification, the preclinical development, hopefully leading to clinical studies, and then hopefully at some point leading to commercial products on the market. And especially in the later phases, in the clinical trials, and also in the approval phase, there are quite a few challenges connected to analytical assays. Just to name here a few, costs are always a main challenge. Also, shortening the vein-to-vein time is usually a main challenge. When we think about our analytical assays, also regulatory requirements are putting quite a pressure on. And also from a biological point of view, we are working here with a living drug. So we really need robust assays that can deal with this kind of drug substance. So what should be kept in mind when thinking about your IPC and QC? So you should definitely start thinking about it very early. So when you're thinking about your manufacturing, already think about your IPC and QC because both need to go hand in hand. And definitely the IPC and QC part should not be underestimated. So we really need robust and reliable assays for analyzing this living drug. Also, the regulatory requirements need to be kept in mind. And the costs for the IPC, QC, and also the labor time should definitely not be underestimated because it can really add up. And when we come back to this figure here with the analytical assays in different phases, we can also say that there are different requirements for the assays in the different phases. So what we always want are, of course, reliable assays and fast and easy workflows. But then when we think about flexibility, this is something that we really need in the discovery phase. But then later, when we reach the clinical phases, we don't need the flexibility anymore. We rather want standardization. When we think about the different methods that are being applied in different phases, so of course, there are really a wide array of different assays. So microscopy, for example, qPCR, flow cytometry, ELISA, cell culture, microbiology, just to name a few. It's definitely not a complete list. And we can also not touch all of them now in this section. We will just touch 3 of them. So microscopy, flow cytometry, and cell enrichment for genomic analysis. So starting with microscopy, so what's very important in the research, but then also later again in the immune monitoring is the analysis of the tumor microenvironment. And for that we have 2 microscopic solutions. One is the Maxima. With that we can really characterize TME components and showing their spatial distribution, and we can really analyze over 100 of markers in the same section. And that's really interesting to identify tumor-specific target candidates and then also to see how certain drug products really work, if they are really effective or not. With our ultra microscope, we have different versions of it. We can do section-free 3D histology analysis. Again, we can see how exactly our cell therapies are working in a 3D environment, which is also really interesting for sure. Just to show an example here on the left for the Maxima, so this is a nice poster that you can also find on our webpage. So there they used the Maxima to evaluate target markers in a pancreatic adenocarcinoma. They also used it to analyze the CAR T cell infiltration, and then also later if the target marker really remains in the tissue or if there's a downregulation. And also, they used the maxima to see whether the target marker that they wanted then to focus on is also expressed on healthy tissue or not. Then for the ultra microscope, I have here a nice video for you so that you can see it in live. What we can see here is again a pancreatic carcinoma, and in pink are CAR T cells, in green the tumor cells, and in orange the vasculature. So there you can really see in a 3D model how the CAR T cells infiltrated the tumor and how the CAR T cells are interacting with tumor cells, which gives you some more in-depth knowledge about your tumor microenvironment. Then I would like to move to the next topic, which is cell phenotyping done by flow cytometry. And we have our MaxQuant family for that, which can be used in all different phases from research really to immune monitoring. And for the different phases, we have our different models, all of them having different benefits. We also love to support you with our protocols. So for the research and preclinical phase, we have our Miltenyi Biotec tested panels now for a lot of different applications already, CAR T, TIL, Treg, and so on. And you can just check it out on the webpage. And then for the later phases, for IPC and QC immune monitoring, we have our special protocols. And there we have, for example, some for CAR T cell manufacturing, which you can also find on our webpage. If we're thinking about the later phases, IPC and QC and immune monitoring, we would highly recommend our MaxQuant 16 for that, because it really offers the best features and benefits for this phase. When we think about reagents, of course, we have your challenges in mind as well. So what we developed is our reaffinity technology. So we have designed recombinantly generated antibodies, and they have a mutated Fc region to really minimize the background. And they all have one universal isotype, so make the isotype controls easier. And for the research and preclinical phases, we would highly recommend our reaffinity single antibodies because they give you full flexibility in your panel design, and you can combine them with your CAR detection reagent. For your clinical phase, we would highly recommend our StainExpress dry antibody cocktails and also our CAR detection reagents. So for those of you who don't know our StainExpress dry antibody cocktails yet and don't know our CAR detection reagents yet, let's dive a bit more into detail into them. So our StainExpress, those are dry antibody cocktails. So you have one tube with all the antibodies in there already, and you just need to add your sample. They are really easily stored at room temperature for 1 year, and you can flexibly add your CAR detection reagent, your reagent of choice, to the panel. And we have already 2 panels available there, and of course we are working on more. On the other hand, we have our CAR detection reagents, and we have already quite a few available there, targeted against CD19, BCMA, CD22, and CD33. And for CD19 and BCMA, we know that they can detect all the commercial CAR T therapies. And just for your interest, we have 2 ways how we detect the CAR. One is antigen-based, so part of the antigen is included in the CAR detection reagent. And we also have another method, which is an anti-idiotype detection method, which is then very CAR specific. And this is some data of our CAR detection reagents. So we tested them on CAR T cells, which you can see on the left side. So we tested there our CD19 CAR detection reagents, our BCMA, CD22, and CD33. And of course, they are not cell type specific, so you can use them on any kind of CAR cells. On the right side, for example, you can see CAR NK cells, where we tested the CD19 and the CD33. And as mentioned before, we really wanted to minimize unspecific background, and especially in the CAR detection reagents, that makes completely sense. For example, for patient monitoring, when you have really a rare CAR T cell population in the blood, you really want to reduce your background. And what you can see here on the left is a healthy donor. So this person never got any CAR T cells. And you see in panel A, no CAR detection reagent was added, so very low signal. On the right side, you see our BCMA CAR detection reagent added, still very low signal. And then on the lower panels, you see CAR detection reagents from two other vendors, and there's quite some high background. So this is something really to keep in mind, that you should work with reagents that have really a low background signal and don't give you false, positive results. Also, we would love to support you with our software to really standardize and automate your workflows. So we have there, for example, our Express modes. They are doing fully automated your gating and analysis for you. So what we have done there is that we have analyzed a lot of different samples, up to 1,000 samples, and built an algorithm that then does the gating completely for you. So not only the gating strategy, but really decides where to discriminate between populations. So there that you can really reduce human biased decisions. We also have our smart gain technology, which is our software to really have comparable results from different devices and maybe different manufacturing sites, so that you have really a multi-instrument alignment over maybe different continents. What we also offer is a software add-on, which is called 21 CFR Part 11 compliant software. And that includes then an audit trail, so that you can always check and document what has been done. And it also includes a user management. To sum this up a bit, so what we would recommend is a complete package for your IPC and QC and also immune monitoring, to go for a MaxQuant 16 system, use the CAR detection reagents together with the StainExpress cocktails, and let the analysis be done via our Express modes. And we already have 12 panels available for CAR T cell processes, and we are also working on panels for the CAR NK manufacturing. And this is an example data set. So this is cells stained with our CAR detection reagents and StainExpress, and gated and analyzed with our Express modes. So what it does in this panel, it excludes the debris, it gates on the leukocytes, excludes then the dead cells, gates on the T cells, and then checks for CAR frequency, and then still checks how many of the T cells that have the CAR are CD4 or CD8 positive, and also looks at that ratio for the CAR negative cells. And as we know that monocytes are quite a challenge in our manufacturing, we also have a monocyte contamination gating there. So this was just an example of one of the 12 panels.
Thank you, Melanie, and hi, everyone. All right, so next I would like to present our solution for magnetic enrichment of CAR-expressing cells. As indicated on this bar on the left, this kit is especially relevant for the preclinical research and also the immune monitoring phase. So the Max Prep CD19 CAR microbead kit can be used for the isolation of CD19 CAR T cells from whole blood. And this is especially useful for downstream flow cytometric analysis with increased sensitivity, for in vitro functional assays, for example, co-cultures with target cells, or genomic analysis, such as gene expression analysis. The kit is compatible with the commercial CD19 CAR T products on the market, and the protocol, unlike other conventional protocols, does not require density gradient centrifugation. The kit includes buffers for erythrocyte aggregation and sedimentation, and the staining reagents for the subsequent labeling of the CD19 CAR engineered cells. So first, the CD19 CAR is recognized by the CD19 CAR detection reagent biotin, with which you are already familiar now. And then magnetic antibiotin microbeads are employed to allow for cell separation. Our microbeads are nano-sized and the smallest beads on the market, actually. So in combination with a high magnetic gradient in our MAX columns, this technique requires minimal labeling and is compatible with all downstream applications. So to achieve CD19 CAR T cell isolation, the column is placed in the magnetic field of a MAX separator, as you can see here. And by running the cell suspension through the column, the CD19 CAR cells are retained, while other cells can be washed away easily. So then the CD19 CAR cell fraction is eluted by removing the magnet. There's also an option to automate the separation process with our AutoMark cell separator. Here you can see an example of CD19 CAR cell enrichment. So in the upper right quadrant here, you can see the CAR positive viable T cells. And before enrichment, they constituted 4.21% of cells. And after the separation, the purity of the suspension is increased up to 96.67%. So this shows that a high purity can be achieved of CD19 CAR cells by using this enrichment kit. With longer time periods since the infusion of the CAR T cells has taken place, the blood can really have very low frequencies of residual CAR cells, as is shown in this example here, where it is only 0.27%. So in this case, after separation, the percentage was increased up to almost 80%, and this can be even further increased if needed by using a second column. So even with a low frequency starting material, a high purity of CD19 CAR cells can be achieved. Next, I would like to talk about functional profiling of cells, which is a major challenge during quality control. But nevertheless, it is really indispensable to obtain reliable and reproducible functional analysis because it's crucial to predict treatment efficacy. Okay, so let's see how we suggest to do that with our solutions in the next slides. So there are many different ways to investigate functionality of CAR T cells, and one of them is to set up co-culture assays with antigen expressing and antigen negative target cells. So by encountering an antigen expressing target cells, the CAR T cells will respond by proliferation, the secretion of pro-inflammatory soluble factors such as interferon gamma, interleukin 2, TNF alpha, perforin, or granzyme B, for example. And this ultimately can lead to target cell lysis. And so, as you notice, there are many different readouts for functional assays. And the one I would like to focus on today is the cytokine response. And I want to introduce two of our solutions for flow cytometry, which enable automated acquisition and data analysis with our MaxQuant analyzers. So the first detection assay is the MaxPlex cytokine assay, which, as displayed on the bar on the left, is most relevant for research and preclinical assays, as well as IPC-QC during manufacturing. So this kit allows you to analyze up to 12 secreted cytokines in parallel. It's a multiplex bead-based assay, which is compatible with standard flow cytometers. It is truly quantitative, so standards of known cytokine quantities are provided with the kit. And if you don't want to analyze 12, but let's say 3 cytokines of interest, you can mix and match a smaller individual panel. Yes, so we have also solutions for human and mouse samples. And we have the express modes to automate your data analysis, enabling unbiased gating decisions. So let's take a brief look on how this assay works. So as you can see here, the beads are coupled to anti-cytokine antibodies, and the cytokines are bound by these specific antibodies, and then they are labeled with APC conjugate antibodies for quantification. Okay, let's take a look at an example experiment. Here, the supernatant from co-culture experiments of CAR T cells and target cells was analyzed using the MaxPlex kit. And as you can see in the mock control and in the cultures with a non-functional CAR, no or very little cytokine release was detected, whereas high amounts of interferon gamma, IL-2, TNF alpha, and GM-CSF were observed upon CAR T cell activation. So this data, which is already published, you can also find in this application note on the right for more details. The second possibility to assess the cytokine response is the rapid cytokine inspector kit. So similar to the MaxPlex kit, the cytokine assay is also most relevant in the research and preclinical phase, as well as IPCQC during manufacturing. So this kit allows you a combined surface marker and intracellular cytokine staining in one experiment. It's a fast and easy solution for activated T cells. It contains an antibody cocktail, so no tedious pipetting of single antibodies, which could introduce errors and variation is needed. It also comes with Brofellin A and all the reagents for the fixation and permeabilization of cells. And then you can just choose your anti-cytokine antibody of interest, which has been optimized for usage in combination with this kit. We have the solutions for human samples and, of course, the express mode for the automated analysis. As you can see here, through the addition of Brofellin A, the cytokines cannot be secreted and remain within the cells. And this is why they can be stained intracellularly. You can choose between a CD4, CD8 T cell detection cocktail or CD4 or CD8 single solutions. So here again, some example data. You can see the staining of interferon gamma, TNF alpha, and IL-2 in the PE channel. And you can see that the amount of cytokines inside of the cells is increased in co-cultures with antigen-expressing target cells compared to the ones with antigen-negative target cells. And of course, a CAR detection reagent can also be added in the panels in one of the 3 channels. To sum up, here you can see again the introduction slide with the requirements throughout the CGT drug development process. And as you can see and probably know from your own experience, some needs stay the same and some change as you progress in the clinical trial. So you will always need reliable assays and fast and easy workflows. Whereas in the early discovery phase, you will need flexible assays to answer different scientific questions. But as you progress in the clinical trial phase, highly standardized assays are absolutely required. And the products we have just presented to you are designed to fit exactly these needs. So we have the right imaging tools for target discovery, as well as our reaffinity recombinant antibodies, which you can use to design your individual highly reliable panels, as well as our ready-to-use kits and antibody cocktails. And for the highest standardization needs in flow cytometry, the full workflow package with the MaxQuant and the express modes, our StainExpress dry antibody cocktails, and the CAR detection reagents. And with that, I would like to thank you for your attention and participation in this webinar. And if you are interested to learn more about our solutions, on our CGT Analytics web page, you will find some more detailed information and also a lot of products that were not presented today due to the time limitations. We also have compiled some interesting material for you about the tumor microenvironment, our CAR detection reagents, and the express modes. Feel free to reach out to your local sales organization, and thanks again for your attention.